Welcome to day 7. Um, Last time I told you that I was gonna go over the next part after the nucleus, but yesterday while I was searching on the news, I discovered an innovative kind of technology that has been unveiled by Korea's Advanced Institute of Science and Technology, and while it is also interesting, it is not only interesting, but it contains some content I talked about, which is protein synthesis in these processes. And most importantly, it's cancer treatment. It's biology. So today I'm going to talk about this, and we're going to cover the next originally planned content in the next video. Alright, so explaining the background, KAIST University, which is a university in Korea, have discovered a way to cure cancer, and it is not curing cancer by killing the cancer cells, but regressing uh, the cancer cells again to normal cells, and which thus not killing the cells and just restoring their normal functions. Right, let's delve into it right away. First is the differentiation process. So in this process, scientists figure out how cells differentiate. Differentiation basically means, so considering protein synthesis, the genes sometimes turn on, which means producing proteins in certain situations, but sometimes they do not. And scientists figure out these processes to figure out the whole differentiation process in each gene, when it turns on and when it turns off. Next up, they figure out the transcription factors, shortened as TF. Transcription factors. Transcription, we have heard this terminology before, and I've talked about it in protein synthesis. So it is basically the starting step, a milestone for the production of proteins. They figured this out by looking at the differentiation process, creating patterns when genes turn on or when genes turn off. When they figure out these transcription factors, the TFs, they basically control the whole DNA synthesis process. Therefore, paving a huge way of relating it to cancer, because cancer is all about mutating and uncontrollable DNA growth or cell growth, which also leads to DNA production. Once they all combine these information, the scientists simplify them to binary numbers. Binary numbers 1 and 0. 1 means the cell is on, 0 means the cell is off. So yeah, it is easier and more simplified and brief to know if the cell is on or off. It's the most concise way. Next up, they combine all these info using an algorithm called a Queen-McCluskey algorithm, and create like a map. This gives like a whole information on, depending on what transcription factors the cell turns on or the cell turns off. And they make predictions based on this algorithm. Next step, using a Boolean GRM model, which is not a thing we are also familiar with. You can basically tell by differentiation process that scientists figure out in what situations the cell turns on or off using this model too. It's basically a next step uh, from the Queen-McCluskey algorithm. So they are all about predicting the patterns of how cells develop or produce proteins or mutate. And therefore, we can discern if a cell is turning into cancer or not. And next up, step 7 is the most important process. Identification of feedback verdict sets. These are also called feedback points. And these are sort of like switches that can turn on the function of a gene or not. So once they identify these feedback points, let's use an analogy of a switch. If a switch is turned up, the whole system goes up, so lights up, or turns on, or functions, whatever. If the switch is turned down, the whole function dies. So it is just like this switch analogy. Now the doctors can manipulate these feedback verdict sets, also known as feedback points, to control the genes. If the cell is mutating and cancer is forming inside our body, we can flip this switch to turn off the cancer and restore the cell's normal functions. This is the most important step. So it's all related to these two factors, differentiation and transcription factors. And the last step is the simulations and perturbations. Simulations, I think you know what it means, and perturbations means like changes, small changes. Changes. These simulations and perturbations are somewhat inflicted among the genes or other predictions that the scientists make to discover the most effective therapy method on cancer cells. This is basically it. So I want to talk to you more about these transcription factors, as it is really related to protein synthesis, which we'll learn more deeply in our school curriculums. So why this transcription factor controls all the process of the gene, and why scientists are really frantic about finding these transcription factors is because in transcriptions, mRNA is moved from the DNA to start the process of making protein, and those process after transcription is followed by regulation and translation, which are the finishing steps of making proteins. So this transcription factor is really important for the gene to produce proteins, and it's a really important way for cancer cells to basically infiltrate. So this is kind of like the independent variable, you know. Finding this makes us figure out the feedback verdict sets, which help us identify if the cell is mutating in a weird way, such as cancer or other tumors, thus making us flip the switch or the feedback points to cure it. So I basically think of this method as, I call it as CRM, cell regression method. So regression is basically going back to the past, and cancer cells regress to the past in order to become normal cells again. This is actually an innovative way, a genuinely innovative way to cure cancer. However, I think it would face some challenges because you see mutations can mutations don't always stay the same. There are always new mutations to form new kinds of cancer or new kinds of appearance of a cancer cell that this process might not work. So let's say we created a map based on all these predictions, all the past cancer mutations that happen. However, what happens if a new mutation just happened and it's completely different from the predictions all the scientists made? Then we have to make a huge change to the map as the differentiation and transcription factors change depending on the mutation. However, it is still a great method as it is not killing the cell but regressing the cell by it is just healing the cell, not killing it. Anyways, this is all for today, and I hope cancer will be cured in the near future, and I'll see you in the next video. Thank you.